Hi, welcome back to Section 2. Why do we need normalization? So there's lots of variation in gene expression values that can occur on microarrays as well as RNA-seq. And some of the variation we're actually interested in. So this is the variation caused by treating samples in a different manner, giving one a particular drug and the other one not the drug, or it might be differences in the disease state. Um, this is the signal that we're after. So we might be interested in what is the difference in gene expression levels between acute myeloid leukemia and acute lymphoid leukemia. And that's the signal we are after. Unfortunately, there's particularly in the high throughput omics data where we try to measure 25,000 genes at the same time, there's a lot of variation which we're not interested in. This is caused by, let's get a pointer. This is caused by the chip spotting process, different size spots, for instance, sample handling and labeling, quantity of sample applied to the microarrays, hybridization time, and the temperature of hybridization, image scanning parameters, image processing, and the list goes on. There's huge amounts of variation that is not the signal, it's not the biological difference between, it's caused by the measuring device and the way we process the samples. This largely falls into two types, which you're probably familiar with, but it's good to define. Noise is the random variation, potentially from the true value of a signal, caused by, for instance, just random changes in temperature over different sampling periods, or random aspects to do with image scanning. So typically this results in Gaussian or normal distribution around a mean value. And we can minimize the noise level by doing replicates. So if we take 5 replicates of a biological condition, measure them, and then take the mean value, then that's going to be a more accurate value than any of the individual measurements. So that's noise. But another issue is systematic bias. So this is where the value is actually, in a sense, calibrated incorrectly. So it doesn't matter how many replicates you take, what you'll get is a more accurate value of something which is got a bias. It's away from its true value. So this is the sort of thing that could happen when you've got two different dyes and one dye fluoresces more intensely than the other dye. Then that first dye is always going to have intensity values which are slightly larger than the second dye. So this is a systematic bias. Using one dye will always give you a higher value than using the other dye. So noise we try to minimize using replicates and experimental, making the experimental procedure more uniform. Systematic bias, this is what normalization is all about. It's trying to minimize the systematic bias while retaining as much signal as possible. So that's why we need normalization. And it's good to see some examples of this because so we've got some data here, and this is a spotted microarray experiment because we've got ratios of CY5 to CY3, and we've got the amount of genes that fall into that ratio bin. So we've got 0 to 1, 1 to 2, 2 to 3. And you'll see that a lot of genes have rough ratios of falling in the bin of 0 to 1. A few are 1 to 2 and then 2 to 3. These ones are twofold upregulated compared to these ones. We've got sort of two samples here where we potentially have a reference sample given as the CY3, and this represents genes which are upregulated twofold, or 2 times the reference level of the gene. This is 3 times the reference level of each gene in turn. So what's this then? Is the genes that are unchanged? Well, some of it will be. A lot of its genes do not change in an experiment where, say, you add a drug. You may only get 100 genes changing, where the organism might have 2,000 genes. So a number of them will have ratios of 1. But actually, also in this bin, we have ones which are downregulated twofold. They have a ratio of 1/2, and those that are threefold downregulated, it's got a ratio of 1/3. So actually hidden in here are all of the downregulated genes as well. So one of the key things we have to remember to do is take these raw ratios or raw expression values and apply log base 2, and this converts it to genes which have a value of ratio of 1 will go to 0. Those that have a ratio of 2 will go to +1. Those with 4-fold upregulation will be +2. And those that are downregulated twofold, so a value of 1/2 will be -1, a value of 1/4 will be -2. So we get this log ratio, and it's log base 2 is what's traditionally used because it's what bioscience researchers are used to, that +1 is a 2-fold upregulation. It's on its natural biological scale. A lot of genes have 2-fold upregulation, they have 4-fold upregulation, less 8-fold. So it's a natural scale for gene regulation. It also has a key purpose as well, which is to make the data more Gaussian-like, more normal distributions. As you can see, this is very skewed, the ratios themselves, whereas this is sort of normal-ish in its distribution, which makes statistical testing using hypothesis tests more, makes them more reliable, because that assumption of a parametric test that is a normal distribution is satisfied, or at least sort of satisfied. Okay. So we could have an experiment with 2 samples where sample A had the CY5 dye, which is a red dye, and sample B had CY3 dye, which is a green dye, and we take the ratios, and this is showing that yes, in this sample with CY5 these genes are 2 times upregulated compared to these genes in CY3, the equivalent genes. So that would be a pairwise experiment, but most real experiments involve more than just a pair of samples, and in that case we quite often have the CY3 as a reference sample. So it's a sample that everything is compared against. So if in that case, if we have 2 samples, A and B, we would have the sample A against the reference and the B against the reference, and we would be wanting to compare things. Again, we might have sample C and D, it might be a time series of lots of samples. But when we do that, and we take the log 2 expression ratios, we could get this scenario. So we've got one microarray giving you this orange distribution, which is largely centered about 0, but this other microarray has given you this blue bars, and it's centered roughly about 1. So this would indicate, because the complete distribution is shifted, that largely all of the genes in sample A have increased twofold, because it's +1 log 2 increase. They've increased twofold over the genes in the orange sample. So that's actually very unlikely in terms of biology, because most things we study perturbations, small changes, or diseases where skin cells and then skin cancer cells, you wouldn't expect all of the genes to increase in expression. You would expect a subset of them that are involved in the cancer. So it might just be 5% of the genes or 10%. Or if you give a drug to an organism, you might expect a small number of genes to change. So this global change is more likely due to systematic bias. Possibly someone has accidentally pipetted twice the amount of sample onto one of the microarrays, or maybe the time it took to do the experiment, the temperature had increased and the hybridization is stronger at a higher temperature, so all of the intensities on this second microarray are larger. So these are the sort of differences that we handle by applying normalization to get rid of systematic biases. Tries to eliminate. And one of the simplest normalization approach is simply mean or medium centering. We've got one sort of distribution here and one distribution here, and we simply subtract the difference from all of the genes in this sample. Now that seems like you're cheating somewhat, but the reality here is that it's assuming that on average the gene expression from one sample to the other will be largely the same on average. So you might have 10% of the genes changing, but you don't have 100% of them changing. And you would also expect genes to be downregulated and upregulated. So with 10% changing, you would only expect a sort of 1/10 of the area of the genes under these curves to actually have shifted. So a gene that was here might have shifted here, up regulated to here, or down regulated to here, but you wouldn't get complete distributions changing. So by normalizing it, you're saying on average all of this sample data values should on average be the same as this sample. And once you shift it, then what you're interested in are genes which actually change position within this sample compared to that sample. I think this is maybe easier to see. So that's spotted microarrays, and that's complicated by the fact that we've got 2 channels. But with Affymatrix expression, we only have 1 chip and 1 channel. So if we actually take exactly the same sample, self against self, and we put it onto 2 Affymatrix chips, expression 1 log 2 ratio and expression 2 log 2 ratio, if we were in an ideal world with perfect measurement, all of the data points, so each point represents 1 gene on the Affymatrix chip. All of the data points should lie perfectly along this orange line. So say actin, I always use actin and myosin being a computer scientist. So the actin gene, it might have in sample 1 the value of 10, then it should also have a perfect value of 10 in sample 2, and myosin might have a value of 12, and it should also have 12 in sample 2. But what we see here is both random noise in the genes, self against self, so they should have identical values, but we've got this much wider bar indicating there's a level of noise. In fact, there's some asymmetric noise, and this chip 2 has measurements of this gene, for example, which on average are much higher than what's been measured in chip 1. So we've got this noise pattern, which we can only have replicates to minimize by taking averages, but we also have systematic bias. It starts off roughly the same, 8 and 8, 6 and 6, but it slowly drifts off. That chip 2, or chip 1 in this case, is measuring a slightly higher value for the same gene as chip 2. So for instance, this gene, or these genes, which are roughly on average have that value 12 in chip 1, they've got values of 11 on chip 2. So this is systematic bias, and it's intensity specific systematic biases. As the genes get higher expression, the two chips deviate more between them. How can we handle that? Well, we can first show this better on the M versus A plot. So this stands for minus, it's one chip log expression minus the other chip's log expression, which is the same as the log expression ratio between the two chips. So if they're identical, then it should be 0. If one's upregulated, it's +1, upregulated 2 times. If one's downregulated 2 times, it's -1. So that's the transformation we use. We use the log expression of one chip minus the log expression of the other chip, and this gives you your M value, your minus value. Whereas the average, the A, is simply the sum of the two chip log expression values divided by 2. So if we look at, for instance, this point here, it's got an average expression of about 8, and the two chips would give you exactly the same expression. But this point here, it's got an average expression of about 8.5, but one of the chips has got a value which is almost +1 more than the other chip. So how do we get rid of this systematic bias? You can see one chip is generally giving lower expressions than the other. We can fit a low S regression. That's a locally weighted smoothing line, and it fits like this, and it's intensity specific bias it gets rid of. And then we take the average at that point and take that away from all of the genes in that window, and in this one you take that difference away from all of the genes in that window, and here we take this difference away from all the genes in that window, and essentially the low S gets rid of this bias, and we get on average very similar on average values within all of the different intensity windows. Now again, you might think, well, this is slightly unfair. One chip was reading values which was less than the other, and we've just got rid of it. But we have to remember that this is a self against self experiment. So the two samples were identical. So it does actually show you how much systematic bias the measuring device has, and that this is actually quite an effective way of getting rid of this bias. And what's left is the noise, because in theory all of these points should be lying along this zero line. And what you would get in real data is you would hope lots of points are lying along the zero line. They're unchanged with the noise level to some degree, maybe up to 0.5 in this case. And then real genes, which are fourfold upregulated, will be standing out here at +2, or if they're eightfold upregulated, they would be at +3. So you would detect the signal, real genes which are changing, very much above the level of the noise. But this is all relying on the majority of the genes will not change. That, say, only 10% of the genes actually do change up and down regulated. Then you can apply this. If 50% of the genes change, this would be a very bad idea, because you actually lose that information. If 50% of the genes all go up regulated, for instance, this normalization would start losing the signal. Okay, so that's possibly a simple example of intensity-specific bias that we've got rid of. But spotted microarrays and high-throughput omics techniques in general, there are lots of biases. So for spotted microarrays, there are dye biases. Green and red dyes fluoresce differently. There are spatial biases due to the way that the microRNAs washed across the surface, and actually the washing of the chips. There are fin tip biases due to certain pins being slightly damaged. So often we want good experimental design to try and eliminate these biases. So for dye biases, we can do dye swapping. We can do one sample is green and one sample is red, and then we can switch them over and then average them, and we get completely rid of the dye bias, and it doesn't need normalization to get rid of it then. So good experimental design is always important. You may be thinking, well, you don't do spotted microarrays anymore, but these ideas apply to all omics high throughput data. We have to worry about biases, and we also have to worry about how to normalize data correctly. It's just spotted microarrays and Affymetrix are good examples where you can demonstrate this easily. So RNA-seq, using next generation sequencing machines such as the Illumina HiSeq, which essentially counts the mRNA molecules by sequencing small sections of them. So it's a discrete measuring device, which makes it much more sensitive and accurate than analog devices such as the microarray. So this is me at the Sanger Centre, where they have hundreds of next generation Illumina sequencing machines. They made me wear a blue jacket to say that I was a computational person. Everyone else was wearing white jackets, and I think this is to tell everyone else that I shouldn't be sort of playing around with any of the machines, because I was a computational person. So it's RNA-seq. The basic process of RNA-seq is we start off with the messenger RNA. It then gets fragmented into sections. Then small adapter molecules are added, and that allows it to bind to something called a flow cell. And this then, if you watch the video, so I've done there's a couple of videos about this on Moodle. It's a sort of basic video and a more advanced video about how Illumina works, and it's worth watching those. Basically, it forms, these bits of DNA form little bridges which amplify them into little dots of the same sequence, and then they're sequenced similar to Sanger sequencing, and they form all of these reads. But because there are millions of dots, you can sequence millions of fragments of messenger RNA all at the same time. And then the computational aspects, you take all these fragments and you align them to the original genomic sequence. And the key thing is how many counts you get, or how many reads you get aligned to a particular gene, because that gives you a sense of how much of the mRNA was actually in the original sample. So if you've got twice the amount of messenger RNA, you expect to get twice the amount of fragments being sequenced, and so you would get twice the number of counts of fragments that are aligning to that particular gene. So that's the basic principle there. Yes, I should disappear so that you can see the bottom of this slide. So this shows you a basic workflow of how that data is processed. The input here are the FASTQ files from the Illumina sequencing machine. So these are a bit similar to FASTA files, which David would have talked about, but they also have a quality score. So we've got a sequence, a nucleotide sequence, but each of the nucleotides has a quality of how well it's been sequenced. These are fed into packages like Bowtie or Top Hat, and they then align these small fragments against a reference genome. So if it's a human sample, this would be human genome given as a FASTA file. And this is quite intensive because there are tens of millions of these fragments needing to be aligned. So there's new techniques to do that. And then one step on, what we want to do is count how many fragments or transcripts or reads are aligned to particular genes. So this is something that a software like Cufflinks does. It takes in a gene annotation file, GTF file, which gives the position of the genes on the genome, and Cufflinks then counts how many reads or fragments or transcripts, they're all called, are aligned next to that gene. So we get a measure of that. Transcript levels are given in RPKM. Reads, so each sequence read, per kilobase of transcript per million mapped reads. So this is a sort of normalized value. So if the gene is the same expression, but the gene is twice as long, you would then expect twice the number of reads. So this is why it's reads per kilobase of transcript, per kilobase of how long the gene is. If the gene is 4 times as long, you'll expect 4 times the number of reads. So it needs to be normalized against the length of the gene itself. And then the per million mapped reads, it's because if you run the Illumina flow cell, it may give you 2 million reads coming from it, and then another run of the same sample may give you 4 million reads. So what you would expect is then the genes will have twice the number of reads compared to the fast run of the machine. So it has to also be normalized against how many million reads did the machine actually produce that time, because it varies depending on chance as well as experimental conditions. So then eventually you can use software like CUFDiff, which does actually a differential gene comparison between two samples, and we'll come to talk to that about that later. We also have viewers of this, so integrated genome viewer is probably one of the common ones, and this shows a region of chromosome 22, and there's a little red bar showing that particular region from, I think it's 31,000 kilobases to, just slightly long, 31,500 kilobases. And each of these is a mapped read that's shown here, and these are two samples, and you've got a distribution of the density of reads, and you can see that this indicates an exon intron structure. So we've got an exon here, there's a big intron exon, and it's difficult to see, but this structure here is exon and introns being detected. So this is another aspect which is good about next generation sequencing, is it allows you to identify the structures of genes which were actually quite difficult to discover before NGS results. So the final genes are shown here, so you can see the exon intron structure that's coming up in the reads. So no matter what technology we use, biological replicates are important. So if we use spotted microarrays, Affymetrix, RNA-seq, or in fact any omics technology, it may be proteomics with mass spec, or metabolomics with high resolution NMR, we always have to be concerned of having sufficient biological replicates. So in the acute lymphoid leukemia, acute myeloid leukemia study by Golub, he had 27 independent samples from acute lymphoid leukemia patients and 11 independent samples from patients with acute myeloid leukemia. These are biological replicates since they provide information about the variation between biological samples. If he had taken a sample from one patient and put it onto two Affymetrix chips, then this would actually be a technical replicate, because it's measuring the variation in the noise levels measured by these chips. Whereas a biological replicate gives you more information about how much does that gene vary within the biological population. So this is an important difference. You should try to have at least 5 biological replicates, absolute minimum of 3, because high throughput data tends to be noisy. Replicates minimize the random noise by allowing you to take the mean expression values of say 5 replicates. But they also provide information about the variability of the expression value of particular genes in the natural biological population. So if we have, say, cell cultures, and we measure a cell culture derived from different people, we measure the actin concentrations. By having 3 replicates, we get a better sense of what is the mean value of actin gene within these different cell cultures. But that also gives us an idea of what is the variation of different people with different genomic backgrounds compared to the mean value. So actin you wouldn't expect to maybe change so much between cell cultures, but if it was, say, a glucose metabolism gene, and it was coming from, say, mouse samples of liver, then it very much depends on how much that mouse is actually fed when around the time of the sample being taken, because that will affect the gene expression level enormously, how much food it's had. So you would expect far more variation around the mean value for certain genes, like metabolism genes, where diet is not being controlled, compared to what used to be called housekeeping genes, which are quite stable from person to person. So all of this is important to consider. So all of these techniques have different upstream processing. So the spotted microarray, we get ratios between two channels, and we have to take log ratios. With Affymetrix, it's a single channel technique, and we get log expression values. And for RNA-seq, we get counts against particular genes, which we then use statistical modeling to determine, quite often, log expressions as well. So they all require different normalization and different upstream processing, but they all eventually result in gene expression matrices. So this is where we have a number of samples, A1, A2, A3 for instance, being one type of sample with 3 replicates, and B1, B2, B3 being a different sample type with 3 replicates. And for each one, we have the measurement of all of the genes in the organism. So we can see in here, samples A1, A2, A3 have expression values of 6.6, 6.4, 6.3 for gene 1, and these B samples have 4.2, 4.4, 4.3. So it's gone down roughly 2 units, which in log expression value terms would be a fourfold down regulation of the gene. So this is the type of data that we eventually get from transcriptomics data. So what have we looked at in this lecture? Well, we've looked at gene expression data. It's one of the most popular omics data types for machine learning analysis. We've given an overview of spotted microarrays, Affymetrix, RNA-seq, how each of these technologies work in terms of the underlying technology itself, how it measures the mRNA concentrations, but also the type of data you get. So spotted microarrays, you get ratios between two samples, and we use log ratios. Affymetrix, single channel, you get an expression value for each gene, and we use log expression values. And then RNA-seq, you get these counts of fragments of sequence across each gene, but those are often turned into measures like fragments per kilobase of gene per million reads, or with statistical modeling like from Section2 package and bioconductor, we can sort of turn it into log expression values. So it's sort of important to actually understand some of the experimental background to know about replicates, data pre-processing, normalization, because if that isn't carried out correctly in an experiment, the actual downstream machine learning is often quite meaningless. So in the next lecture, we're going to cover actually how to represent this omics data, and particularly how to represent experimental data so that we can actually give the context to this high throughput data in terms of the biological experiment. I've just sort of said that, well, we get a matrix of numbers coming out, and that's a perfect sort of thing that can be used in machine learning. But the reality is that biology has much more data associated with that matrix of numbers than is actually used a lot of the time in current machine learning of omics data. And this all is prior knowledge, and it would be useful to use. So if it's a cancer sample, some of the genes will be oncogenes. They're known to be involved in cancer, sort of in the cause of cancer. Some of them will be tumor suppressive genes, known to be involved in suppressing cancer. And to actually not use that information when sort of prior information when doing machine learning with the actual gene expression values is a real shame, but often it isn't, because people don't fully understand the biology and they don't have a way of representing all of this sample data information and gene data information, all this metadata. So that's what we'll be describing in the next lecture. Okay, so I'll see you then. Okay, bye.